This 3D printed contraption lets a lab automation robot use a pipette to flip a switch. This brings a magnet close to a tube, attracting the magnetic beads with DNA attached, so the robot can then pipette away impurities. This is one of several hacks that biotech company Plasmidsaurus uses to provide sequencing as a service at unprecedented scale and price. The lab behind me is one of 10 Plasmidsaurus labs around the world that together have sequenced 5 million samples of DNA in just the last 3 years. They have spent that time iterating on improving every process, ranging from lab automation to delivering bioinformatics results.
If you're going to do 96 magnetic bead separations at once, you can use a device like this where that's 96 magnets. And then this has a tip inside of it. So we use that this just gets picked up by the pipette, and then we can tell the pipette to engage or disengage the magnet by using the tip eject mechanism. And so you can see there's gears on here, and this all 3D printed except for the springs. And then we tell it to eject, and you can see that causes the magnets to go up. And then if we want to put them back down.
So the whole pipetting mechanism just thinks that this is a pipette.
Yeah, it's telling it's going to eject the pipette, don't eject it.
Their first and main offering is whole plasmid sequencing. If you had your business hat on and did a market analysis just 4 years ago, demand for this service would have looked very low. But Mark and team figured out that they could afford to offer it at such a low price point, $15, that it could compete with the much more commonly used Sanger sequencing that was routinely done to check small sequences within a plasmid.
People started getting the entire plasmid sequence every time. And what they learned is that, yeah, there had been this delusion that the only thing that matters is the part you modified. But there was a lot of parts in plasmids that would go bad over time anyway. So what would happen is somebody would once make this 30 binding site motif in their plasmid. They would do a lot of work with restriction digest and all sorts of things to verify that it was what they thought it was, and then never sequence it again. When we first came out, we got emails all the time from people that were like, hey, this experiment had stopped working. In the worst case, there was a PhD student who had spent years trying to figure out why this thing wasn't working full time. And then she sent it to us and immediately was like, oh, this plasma was wrong. A bunch of stuff got deleted. But you're never taught that, or you hadn't been taught that at the time, that this was actually even a possibility.
So as we drove home from this Plasmidsaurus visit, there was one thing on my mind the whole time. The idea that something is expensive and overkill for most applications, but as soon as it becomes cheap enough, it could replace a lot of other applications or create demand that didn't exist before, because now it's so cheap you might as well do it. We're really entering that era with DNA sequencing in general, I think, where whole genome sequencing becomes a much cheaper alternative pretty soon to even doing things like disease-specific gene panels for doing genetic testing of patients in a clinic. If you can just sequence everyone's entire genome, you don't have to keep going back and being like, oh, now this person has a new disease, we're going to sequence a different gene panel. All the bioinformatics of certifying that gene panel to have the right statistics in terms of sensitivity and specificity, we don't have to do that for every type of disease, for every gene panel. We can instead do whole genome sequencing, and it becomes easier to standardize once we do the thing that is overkill, but we learn to do it really well and really cheaply. We can standardize around it, and it's ultimately going to make everybody better off. Another great example of this concept is, in fact, that Plasmidsaurus is now offering RNA sequencing as a service. They're offering it at a price point, I believe it's now $50, which is cheap enough that they hope scientists will start doing RNA-seq instead of things like qPCR. Just like I mentioned with whole genome sequencing versus gene panels, this is an example where instead of checking just individual genes, you check all the genes. This is more expensive because you need to do more sequencing, but it is easier to standardize. And now that the per base cost of sequencing has gotten low enough, we can just do it all, and that'll actually become cheaper because we can standardize the processes.
I think one thing that's quite unique about us is that because we have strong engineering talent, we homebrew a lot of tools that help us be more effective at sequencing. And so these tools obviously span from automation, robotics, and data automation, getting information out of our instruments and into the cloud and using that to make intelligent decisions quickly. There's other cool stuff too, like software that schedules drivers around the world. And we got to let them know, hey, there's samples in these boxes, take this route, bring it over to the lab. But the heart of what allows us to scale rapidly is our ability to turn manual tasks in the realms of automation, bioinformatics, into repeatable scripts.
When you start to standardize things like whole genome sequencing, whole plasmid sequencing, and RNA-seq, you can actually also start to standardize the bioinformatics that comes right afterwards. And I'm excited about this because for years, people have been asking us on our channel to make tutorials for how to do RNA-seq. And I've been baffled that this has not been standardized enough that they could just plug their samples in somewhere and it'll just do it for them. But of course, the cost of having bioinformaticians go and help you with your specific samples is often too high. And so wet lab scientists find themselves looking for tutorials online, trying to do RNA-seq themselves. But when Plasmidsaurus has now taken on RNA-seq, they're also doing the bioinformatics for you. And we talked to some of their bioinformaticians about what they do and how they make this whole service integrated from wet lab to bioinformatics.
RNA-seq is a great starting analysis if you want to get into it, and I like strongly encourage people to dive in and play with their tools. But it's also a barrier, right, to your science and to getting stuff done. You do the mapping and all the quality control and make sure that the data you're getting is really good. You get BAM files back, you get a count matrix, so you're able to take that and do your own analysis if you want. But then on the website side of things, we offer a pairwise comparison analysis.
Another concept that has caught our attention is the as-a-service model. Most companies serving biotech labs sell products like machines that are then part of a protocol that a scientist follows. And labs might not need these machines very often. In our podcast interview with Mark, we gave the analogy of a bread machine. It's like saying everyone needs a bread machine in their kitchen. Exactly. And like eventually your entire counter is just filled with like machines for every possible thing. But I think most of us prefer to just be able to go buy bread at the grocery store. I'm really curious if any of you have ideas for other areas of biology where there's some expensive machine in the lab that you don't need if you can just send the sample out and have somebody else do it for you. Is it an expensive machine? Is it a long protocol that takes many hours of work? Is it a machine that not every lab has? So you don't strictly need it, but it saves you a lot of time if you do have that machine and can do it automatically. Are there any other areas of wet lab biology that you see like this that makes sense to make an as-a-service business out of? I would love to hear your comments on that. And also check out our interview with Mark on our podcast, which you can find here on YouTube or in any podcast app.